Hi, prospective NSP students. My name is Colin Lee. I'm a grad student in the Rhanor Gillette lab, and I'm here today to give you all a tour of our lab. So let's go on in. So we'll start first in 414, one of the two main lab spaces that we use. Come on in. Pan so that you can see the whole thing. You can see the door to Rhanor's office in the back there. And any tour of our lab, of course, has to begin with the animals, so we'll head over to the main observation tank. So in the observation tank here, you can see two octopuses in isolation, and then various critters that are loose in the tank. We'll come over here. You can see one octopus with its tentacles all spread out, probably looking for some food, and another one that's in a hamster ball here. And then scattered throughout the tank, we have various sea anemones, sea urchins, and even one starfish. The octopuses represent a new area of research for our lab, looking at the peripheral nervous system and its role in learning and behavior. The rest of the animals are actually here kind of by accident. They were likely microscopic veliger larvae that were present in ballast water brought into our lab, and then they would have metamorphosed and then grown up here. But so they have become cherished pets in our lab. Over here, we have a second tank with an octopus in it. You can see several cameras around it. This octopus is being used in long-term behavioral studies, ultimately targeted understanding learning. So beyond behavioral experiments, what do we do in the lab? A key thing we do is neurophysiology, and I'll show here an example of one of our rigs. So you can see the rig here. You can see all the electrodes that are around the recording dish. We do both intracellular recordings, in which we penetrate the soma of a neuron with a sharp electrode, and extracellular ones, in which we record from nerves. And the combination of these two methods allows us to develop very detailed understandings of the neuronal circuits that govern behavior. We're now in room 422, the other main room that our lab uses. I'll just pan so that you can see all of it. And the big highlight of this room is this tank right in front of us, which is our new observation tank for the octopus. The goal with this tank is going to be able to ring this entire tank with cameras and get complete coverage of the octopus's movements at all times. In addition to the octopus, our lab does extensive research with the sea slug Pleurbranchia californica. Pleurbranchia CNS has approximately 10,000 neurons with large cell bodies, some of which can be seen by the naked eye. These neuronal characteristics, combined with the animal's behavioral simplicity, make it an excellent organism for the study of neuronal circuits. Here, we show a video of Pleurbranchia attempting to feed on the chemically definite sea slug Flabellina. Pleurbranchia first approaches and bites Flabellina. Upon experiencing its chemical defenses, it then rejects the prey, then turns and crawls away. Individual neurons responsible for each of these behaviors have been defined, giving us an extremely detailed understanding of the neuronal basis of Pleurbranchia's behavior and decision making.
Hello, my name is Katya, and I'm a postdoc in Rhanor Gillette's lab. I focus mainly on computational work, which includes behavior and neural circuit models. For example, using the observed behaviors and circuitry of sea slug Pleurbranchia californica, Rhanor's lab created a simulation of forging decision called CyberSlug. In this model, a virtual slug forges for different types of prey and learns their odor signatures. CyberSlug makes approach avoidance decisions based on what it senses, what it learns, and its own internal states, such as hunger, just like the real Pleurbranchia. We then found that we could easily modify and expand CyberSlug circuitry, in one instance creating a model of addiction called Asimov. By adding some simple wood circuitry with homeostatic mechanisms onto CyberSlug, we were able to simulate some of the hallmark processes of addiction, such as desensitization, withdrawal, and resensitization to higher ward stimuli. We are currently working on further expansions of CyberSlug and Asimov circuitry, including a model for simple episodic memory. In particular, we are interested in bottom-up modeling approaches, which may give insight into possible evolutionary origins of certain complex behaviors.